I'd like to start with my acknowledgments. This project was one that is funded jointly by National Science Foundation and the BBSRC, which is the equivalent in the UK. It's from a special high-risk, high-reward granting program to try to do something out of the box. And we have 4 collaborators on this project. The collaborators that whose work I'm mainly going to be describing are those at Rothamsted Research, the group of Martin Perry and his postdoc Alessandro Ocellini and his associate John Androloj. We also have collaborators at Michigan State. Cheryl Kerfeld has helped us understand something about cyanobacteria, and Steve Long and Justin McGrath at Illinois have been doing some modeling for us. At Cornell, my team has consisted of Miat Lin, who is an NIH postdoctoral fellow. He was assisted by Wenfang Li, who was a technician who's now in medical school. She helped him for 1 year after she graduated from Cornell. And then there are 2 NSF REU students, Kevin Hines and Jenna Rieger, whose work contributed to this. They were here for the BTI NSF REU program. And Kevin has actually come to Cornell and joined the BMCB program as a graduate student, while Jenna has gone on to Penn State for her graduate work. Now, the reason that NSF and BBSRC were interested in a program to do something with photosynthesis that hadn't been done before was the consideration of this problem here. And that is the possibility that we're beginning to exhaust the natural variation for plant breeding in some of our crops. So you can see in the decade ending 2007 that maize is still doing quite well as far as the percent yield increase per decade, but rice and wheat have started to go down, and one of the issues is how can we get those to go back up? There is a major difference between maize, rice, and wheat, as I will discuss later. So the idea is maybe there is some trait that we haven't actually been doing breeding for. It's very difficult to breed for photosynthesis except indirectly. And one problem with photosynthesis is the enzyme Rubisco. The enzyme Rubisco, its properties do limit the efficiency of photosynthesis. One of the well-known properties is that Rubisco can react with either CO2 or with oxygen. And if it reacts with oxygen, then you get photorespiration, which reduces the efficiency of photosynthesis. That's not the only problem, though, with Rubisco. The other problem is that it has very slow catalytic rate. Anywhere from 3 to 10 CO2 are fixed per second. That's relatively slow compared to most enzymes. The other issue is that Rubisco requires a lot of nitrogen from the plant because you have to make a lot of Rubisco since it is a slow enzyme. And so 25% of leaf nitrogen in a typical C3 plant is consumed by Rubisco. So reducing photorespiration then is a target for improving photosynthetic efficiency. If you look at this chart, which was developed by our collaborator Steve Long's group, you can see that the actual carbon fixation is much less than the potential because of the loss due to photorespiration. And that loss increases as the temperature increases. So especially with global warming, although we don't seem to be having global warming outside, global warming does exist, and as the crops are growing in higher temperatures, there's going to be greater loss due to photorespiration. Now, there have been some strategies that have evolved to reduce photorespiration, that have naturally evolved. And these strategies basically are to increase the CO2 near Rubisco. Rubisco evidently has a very hard task to fix CO2. So in C3 plants, we see here that Rubisco is at the mercy of the outside environment. But in C4 plants, there's a mechanism that allows the plants to pump CO2 to these specialized cells in the bundle sheath, and therefore get around some of this problem of the reaction of CO2 with oxygen. There's a lot of effort going on right now to introduce C4 photosynthesis into C3 plants, such as rice and wheat, to make them more photosynthetically efficient. One of the biggest bottlenecks, though, in this effort is the fact that you need to actually change the plant anatomy, not merely change gene expression, but also the plant anatomy. So here is a C3 leaf, here's a C4 leaf, and in order to get this pumping to Rubisco, you will have to do some changes in the leaf anatomy, which is, I think, a more daunting challenge than merely changing gene expression and its location. But this pumping mechanism does allow C4 maize to be a lot more productive than some leading crops. So this is a slide I borrowed from Ed Buckler that he discussed recently. And you can see here that wheat, rice, and maize really are quite productive. They're much more productive than soybean. It's possible that this reduction in the soybean relative to these others is because soybean is fixing its own nitrogen, and there is a cost to that. Nevertheless, it would be nice to have wheat and rice up to the productivity of maize. And one way to do this may be to find some way to reduce photorespiration. So many C4 plants are able to use a faster Rubisco than C3 plants. So here we have the rate of CO2 fixation by Rubisco in C4 plants versus C3 plants. There is some variation in C3 plants and C4 plants, and perhaps this can be exploited as well. But also, you can see that because the C4 plants are able to use faster Rubisco, that's one reason for their enhanced photosynthesis. Now, the problem is, and really the dilemma, is that the faster the Rubisco enzyme, the lower its affinity for CO2. So if we have its reduction in affinity, essentially, on this here, and the rate of CO2 fixation, the faster the Rubisco, the lower its affinity for CO2, and therefore it's more likelihood that it'll react with oxygen instead of CO2. So if we want to use a faster Rubisco, we need to get around this problem. Now, cyanobacteria have gotten around this problem. They're able to use a very fast Rubisco compared to plants. So how do the cyanobacteria do this in contrast to, say, the C4 plants? The cyanobacteria have a carbon concentrating mechanism just like the C4 plants, but it is a different mechanism in that they have these little microcompartments called carboxysomes right here. This is what they look like, these carboxysomes. Here's an enlargement of one of these carboxysomes. The way they work is they have a protein shell, and carbonate is pumped into this protein shell. Then carbonic anhydrase converts the carbonate ion to CO2. The CO2 surrounds the Rubisco, and then the Rubisco can function quite efficiently, even though it is very sensitive to, has less affinity for CO2 than the plant Rubisco. So the other advantage of this cyanobacterial carbon concentrating mechanism is that it also reduces the investment of nitrogen in Rubisco. So while the C3 plants have a very high investment in Rubisco here, the cyanobacteria have a much lower investment of their nitrogen in Rubisco. So one possibility is if you can use a faster Rubisco and need less Rubisco, then maybe you would need less nitrogen devoted to it. So this is the question then. Can the cyanobacteria carbon concentrating mechanism be transferred into plants? And in this very nice schematic shown in a recent Nature News and Views article, you can see the idea here. This is the carboxysome. This is how it works in the cyanobacteria. If we want to get it into the plant, we need to put it into the chloroplast, pack Rubisco in there, and send in, again, the carbonate into the chloroplast and the CO2. So our project then was to try to do this task of transferring the cyanobacterial carbon concentrating mechanism into plants. So how does this work in cyanobacteria? How do you build a carboxysome in cyanobacteria? The information from Cheryl Kerfeld's lab and Pam Silver's lab indicate that Rubisco seems to nucleate inside the cyanobacterium, and then when it reaches a sufficient size, the outer shell proteins of the carboxysome form around the Rubisco. So it does look like first you might need Rubisco nucleation, and then the shell proteins would then surround it. So the first thing one might need to do is to put Rubisco, the cyanobacterial Rubisco, into plants. So how do you do this? Well, fortunately, we can do chloroplast transformation. We can transform the chloroplast genome. And it was worked out by Paul Molliga's lab a number of years ago that you can transform by homologous recombination. So if you have your target gene in the wild-type chloroplast DNA, you can replace this with a transgene by putting sequences on either side that are homologous to the sequences on either side of your target gene. And then after this homologous recombination, you can recover a plant that has entirely your transgene and has eliminated your target gene. Now in our case, we want to eliminate the large subunit of Rubisco, which is located in the chloroplast genome in plants. So this is what it looks like in plants. This is the structure. You have a promoter, you have a terminator, and of course the coding region. And our model plant for these studies is tobacco. The reason we're using tobacco is that it can be transformed the most easily and the quickest of any of the plants that can undergo chloroplast transformation. However, there are some useful crop plants that also can be transformed in their chloroplast genome, such as soybean, for example. But it takes much longer to make a soybean chloroplast transformant. So our idea is to work this out in tobacco, and then once we know how to get this to work, we can put this into some more important crop plants. So what we're doing is we've got on one side we've got the promoter of Rubisco. We put our cyanobacterial transgenes in here, and then we use a selectable marker for spectinomycin resistance that, again, Paul Molliga's lab worked out some years ago. It's the AADA gene here. So this is a selectable marker. And we have to select for spectinomycin resistance in order to know that we have a chloroplast transformant. And this is where in the tobacco chloroplast genome we're introducing our genes. This just shows, for those of you who haven't seen this, a diagram of the generation of transgenic plants that have transgenes in their chloroplast genome. What we do is we start with some seedlings, and we bombard these seedlings using the gene gun with our vector of interest. And after bombarding, you cut the leaves up and put them on a plate that contains spectinomycin. And after about 4 weeks, if you've succeeded in transforming the chloroplast genome, you'll get a shoot. And this shows the outcome of actually a high-frequency transformation experiment. If you can get one shoot per plate, you're doing pretty well. But that's not so bad. It's not that hard to get one shoot per plate if you're experienced. And my postdoc, Miat Lin, is really a super chloroplast transformer and can do this quite readily. Okay, then the problem with this is, while you've got some spectinomycin resistance in there, a typical leaf cell has hundreds of chloroplasts. And so you may not have transformed all the chloroplasts in there. And in fact, you usually haven't in the first round of selection. So you have to take that leaf, the leaves from here, and slice them up and put them back on selection. So they go through a longer period of selection in order to segregate out the wild-type genomes without the spectinomycin and still carrying your large subunit of Rubisco, and the ones that you want. So usually by the second round of selection, we find that we've been able to replace all of the chloroplast genomes with the transgene. But you can test at this point, and if they aren't, then you can go back and do another round of selection. Once you have these homoplasmic plants, they're entirely transformed. That's why they're called homoplasmic. You can put them on a medium to get them to root, and then plant them. They'll grow up, and you'll be able to plant them out, and you've got a nice plant. Now you can see it takes about 3 months, if you're lucky, to make a chloroplast transformant. So it's not a trivial length of time that you need to do this. Now, one question we had is, would it be possible for the plant Rubisco assembly apparatus to assemble the cyanobacterial Rubisco? The assembly pathway has been worked out very well in cyanobacteria. It uses the GROW-EL, GROW-ES system here, chaperone system, to get this, and a protein called RBCX, another chaperone, to assemble Rubisco, because as you know, Rubisco is composed of multiple subunits, a small subunit and a large subunit, and they have to be assembled together properly. Now in plants, the assembly apparatus is much more complex, and that complexity partly results from the fact that in plants, the two subunits are encoded in different locations. The large subunit of Rubisco is encoded in the chloroplast genome, while the small subunit is encoded in the nuclear genome. So you have to get the small subunit into the chloroplast and then get it to assemble with the large subunit. And so there's various chaperonins have been discovered, various assembly factors, and recently the Stern and Barkin labs have discovered some new assembly factors that you need. So it's a very complex process to get this to happen in plants with the plant enzyme. So we weren't sure whether this would work to put the cyanobacterial Rubisco in. And we decided to increase our chances of getting the cyanobacterial Rubisco to assemble, that in addition to expressing the large and small subunit of the cyanobacterial Rubisco, we would also express the cyanobacterial RBCX gene, or we made another construct in which we used this protein here called M35. Now, in the cyanobacterial model here, this is the outer protein shell, and then the green is the Rubisco, the red is carbonic anhydrase. This is the current model of how the proteins are structured or ordered in the carboxysome. So you have the shell proteins here, then you have this protein called M58N, and then that provides a link up here, and then the carbonic anhydrase. But the Rubisco seems to be organized by a protein called M35, which has some sequences on it that are similar to the small subunit of Rubisco. So it's thought that perhaps this small subunit, that there are fewer actual small subunits present in the cyanobacterial Rubisco, because maybe some of these M35s are replacing them. And maybe this is important to get the assembly to occur, and also to get this sort of lattice work here of Rubisco assembled, ready to go to be surrounded by the protein shell. So the constructs that we made are shown here. Again, the selectable markers at the end, but we have the large subunit and the small subunit in each of these constructs. One of the constructs has RBCX gene, the other construct has the M35 gene. The other thing that you need for doing this kind of experiment is you need appropriate terminators and ribosome binding sites, and we're also using something called an intercistronic expression element. What happens is, this is one long transcript here, this is one operon, and this element here allows the processing of this one large transcript into these three transcripts here. So you'll have a large subunit transcript, a small subunit transcript, and the RBCX or M35, along with the spectinomycin marker. Okay, so once we did this transformation, we had to check to see whether the plants were homoplasmic. Do they have only the transgene, or do they still have some wild type sequence? Do this by doing a southern blot. That's what this is down here at the bottom. This is a southern blot here, where we see the wild type DNA is down here. Of course, an appropriate probe was used to find this. And then in the two transgenes, we have the DNA that's the right size for a fragment that's the right size for each of these transgene constructs. And if you look, you can see that there isn't any of this wild type DNA still present. So we do have a homoplasmic plant that doesn't have any of the plant tobacco large subunit. And then you can also look at the RNA level expression. In wild type, if you do RT-PCR for the tobacco large subunit, you can see it in wild type, but it's not present in these two transgenic lines. And if you use RT-PCR for the cyanobacterial large subunit, it's not in wild type, as you could expect, but it is, in fact, present in the two transgenic lines. So this is the bottom line. We were able to get transgenic plants with cyanobacterial Rubisco, and these would grow autotrophically. They are growing slower than wild type. These are the same age, these three plants, but both of these plants will eventually grow and flower and are fertile. They do require supplementary CO2. You would expect this because since there's no cyanobacterial carboxysome in there to provide CO2, if these plants were left just in our atmospheric air, they would die because they wouldn't have enough CO2. And they would, just react with this. Rubisco would react with oxygen. You wouldn't be able to do enough photosynthesis to support the autotrophic growth. And when we looked at these plants to see if we could detect any of the tobacco Rubisco on Western blots, we could not detect any. So we can see here that using an antibody that's specific to the cyanobacterial large subunit, we can see that that's present in these plants. And using a tobacco Rubisco, we can see that it's present in wild type, but not in the two transgenic lines. Having a little delay on this mouse. Okay, so again, we don't see any, using a tobacco small subunit antibody, we don't see any of the tobacco small subunit antibody either, protein either when we react with this antibody. So these plants are actually using the cyanobacterial Rubisco. And the enzyme kinetics in these plants confirms that the plants are expressing the cyanobacterial Rubisco and not the plant one. Now wild type Rubisco is already saturated at this CO2 down here, while with more CO2, the other two enzymes are still going strong. This shows that the enzyme that is present in this plant is the cyanobacterial Rubisco. It has the cyanobacterial Rubisco kinetics. Now, an interesting feature was when we analyzed the electron microscopic view of these chloroplasts. This work was done by Alessandro Ocellini at Rothamsted. In wild type, Rubisco is dispersed through the chloroplast. You can see these little round things where the immunogold has been circled in the wild type, and also in the plant with the RBCX construct. Rubisco is loose in the stroma of the chloroplast. But in the plant with M35, we actually are seeing these large aggregations of Rubisco. The M35 seems to be organizing the Rubisco into a large mass in these plants. This could be similar to the so-called pro-carboxysome that's seen in cyanobacteria by our collaborator Cheryl Kerfeld. If you turn off the shell proteins and have only the Rubisco present, you can see these pro-carboxysomes in cyanobacteria. So this may be a stage on the way to the assembly of a genuine carboxysome. It's also interestingly reminiscent, though, also of the pyrenoid that's present in some algae that's used to concentrate carbon. This again is aggregation of Rubisco, and perhaps some sort of structure like M35 and RBCS ended up evolving into the pyrenoid at some point. So it could be like a pyrenoid, or of course could be merely a procarboxysome. This is what that large structure here that you saw in the EM looks like if you label one of the proteins with GFP, so with YFP actually. So if YFP M35 is labeled, then you can sort of see these very large structures that you see in the EM. You can also see them in this fluorescent image. So to summarize the first part of my talk, don't worry, the second part is shorter. I've told you that we can get an active cyanobacterial Rubisco in tobacco plants. And we can do this with either the chaperone RBCX or the protein M35 if they're expressed along with the Rubisco genes. And we can't find any of the endogenous tobacco Rubisco, so we know these really are performing photosynthesis and they're autotrophic because of the expression of the cyanobacterial Rubisco. And this M35 protein is very interesting because it is causing aggregation of Rubisco into these large suborganellar structures, which may be on the road to making a carboxysome if we can now express the carboxysome shell proteins. So the next step is to introduce the entire carboxysome. A lot of work on cyanobacteria have been done and on the carboxysome, and so a lot is known about what genes you need to build this carboxysome. There is an operon present in the cyanobacteria that we've been using, a strain called Synechococcus 7942. So you have an operon with a large and small subunit, and then these other shell proteins, the internal proteins, and carbonic anhydrase. And even though I've told you that maybe you need a Rubisco first, maybe you don't need Rubisco first, because it is known in this bacterium that you can get a carboxysome, empty carboxysome shells that apparently look fairly normal in shape and composition. If you knock out Rubisco, you don't have any Rubisco, you can still get these shells made in this particular bacterium. So maybe we can get it. Maybe we can get assembly even without the Rubisco. So we decided to try this by using transient nuclear expression by agroinfiltration. The reason we did this is it's much quicker than doing chloroplast transformation. You don't have to wait 3 months. You can wait 3 days, and you can have a result if you do agroinfiltration. So the idea then is you make a vector, put it into agrobacterium, then you infiltrate the leaf with the agrobacterium. It will express the proteins on your vector, and whose genes are on your vector, and then you can look a few days later. So this vector was constructed by Miat Lin to express 3 different proteins. So it has 3 promoters, and it will express 3 proteins from the tDNA of agrobacterium. When he expressed 3 of the proteins that are in the shell, it's known that the shell has CCMO, and it forms a hexagonal structure. The protein as CCML is a pentamer cap here, and then there's also CCMK2 again, which forms a hexagonal structure. So expressing K2, L, and O labeled with YFP, you can see these intriguing little structures inside the chloroplast. Now the red in this image is chlorophyll autofluorescence. So wherever you see red, you have chlorophyll. And you have these little structures inside the chloroplast. When our collaborators looked at this in an electron microscope, they were able to see some structures, some intriguing structures that looked like microcompartments. They're about the right size, maybe some of them are larger, for carboxysomes. So this is a very promising result here. Now this is K2, O, and L. This one is K2, O, and M58, which is one of these internal proteins. And you can also get structures when you express those 3 proteins. You can see there's some sort of strange linear structures here. We do sometimes, probably because the result of not exactly the right ratio of proteins, you can get some strange rod-like linear structures. We can also look to see whether this model holds true in plants when you express these proteins in plants. So it's known in cyanobacteria that K2, O, and N interact in the carboxysome. And if you label N with CFP and O with YFP, you can see that the N and the O are located in the same bodies, indicating that they are able to associate in plants in our transient expression system. Now, it's known that N interacts with the shell protein K2 through a 17 amino acid C-terminal peptide. So that's how it does its interaction. That's been worked out by our collaborator, Cheryl Kerfeld, in cyanobacteria. So does this also happen in plants? So Kevin Hines, during his NSF-REU, deleted the 17 amino acid region of CCMN and found that if he deleted this, then you lost the interaction with the K2O. So here is K2 and O interacting. OYFP is interacting with K2 in order to make these little structures. But N, which is labeled with CFP, is now dispersed in the stroma. It's no longer in these little bodies. So it does need this 17 amino acid interaction peptide in order to work with the shell proteins. So the question then is, could this encapsulation peptide, this 17 amino acid peptide, be used to target a foreign protein to these carboxysomes, to these microcompartments? The reason you might want to do that is you could put your favorite protein into a carboxysome instead of Rubisco. So one thought is, for example, to put nitrogenase into these carboxysomes, except they're no longer the nitrogenasomes or something, because then you might be able to protect them from the nitrogenase from oxygen inside a microcompartment. Or maybe there'd be some toxic protein you'd want to confine to a microcompartment. So Kevin also made a construct in which he fused YFP to this peptide, and he was able to show that after fusing YFP to this peptide, he could get it to associate with the two shell proteins, K2 and O. So that's promising for the possibility of putting a foreign protein into the carboxysome, because we can put YFP in. Maybe we'll be able to put something else in the future. So to summarize part 2 then, I've told you that we can get these beta carboxysome shell proteins to assemble into spherical bodies to resemble these empty microcompartments. And as far as we can see, the protein-protein interactions in chloroplasts are similar to cyanobacteria. And we have shown that this encapsulation peptide on CCMN that works in cyanobacteria also works in plants, and we may be able to then target a foreign protein of some sort to these carboxysome microcompartments if we make them in plants. This last part here was published in Plant Journal, and we actually got the cover. They false colored our little compartments. And the earlier work was published in Nature. Thank you.